Welcome back. So Dr. Ariana Love is back with us again with a horrific discovery. She says that she has a patent proof that COVID PCR tests are actually not tests at all, but that they're linked with human cloning technology. They're not science fiction, are they?
It's real. The PCR kits have never been about testing for COVID, as people were told. It is about cloning. They are cloning devices. And this has been admitted to by the NIH. It's on their website in a study entitled Molecular Cloning, Polymerase Chain Reaction, an Educational Guide for Cellular Engineering. There are patents as well. And the patents do say on the PCR kits, they do say that it's cloning technology. They use that exact word. So here, this study on the NIH website, it explains everything on how the cloning works using PCR kits. So, for example, it talks about vector DNA sequences and fluorescent protein. Basically, all of the kits that are cloning devices, they're using the fluorescent protein. That comes from insects. It's the firefly, the green fluorescent protein. So these are the carriers of the messenger RNA. They get cut into pieces inside the body, and that is used to transfect the human genome. The DNA carried from these microorganisms are then used to change humans. They talk about toxic genes. In some of the PCR kits, they are using toxic genes. They admit to that. And then also they talk about, okay, so what is the testing for? It's for genetic sequencing to determine how far along people are in the cloning process. And you can actually order these DNA sequences online. There's Adgene is offering them, and Thermo Scientific and Biolabs. They literally will send it to you. You order it and they send it to you via email. They're using artificial genetic sequences, right? And they have to delete certain genes in the human genome, and then they code whatever the messenger RNA is coded with, and we know all of them are coded with this green fluorescent protein.
All right, so what is the goal? What is the goal here?
They are changing the human species, attempting to genetically modify humans and treating humans like animals. In particular, the green fluorescent protein, which comes from the firefly, that means they are actually turning people into hybridized humans with insect DNA. So they are transfecting, because we know from the study out of Sweden recently that the mRNA is transfecting and transcribing through reverse transcriptase into the human genome. That means people are becoming part insect. They are going to carry then and insect DNA. So I have clients that have been riddled with parasites, and not only parasites, but also these sort of Morgellons like hydrogels. They're also called Marcellus, and I have a study on that as well. They're laced on the PCR kit. So this is self-replicating nanotechnology, right? And these parasitic organisms are genetically modified, and they can feel them crawling under their skin. I kid you not. And these are people who never took the jab. They just had a bunch of the PCR swabs up their nose. People can go to Adgene. There is type in Adgene plasmid cloning by PCR, type in Biolabs PCR cloning method, and they're going to explain how this is being done.
So the NIH is admitting to this cloning technology.
Yes, absolutely. There's another NIH study about cross-species genomics that I want to talk a little bit about. It's entitled Targeted Genome Editing Across Species Using ZFNs and TALENs. So they have entire genome sequences, it says in the study, available for numerous species. Now, we know they've already been crossbreeding humans and monkeys, humans and pigs, humans and mice in the US, in China, maybe other places, possibly Ukraine, at the US biolabs there. I mean, this is nothing new. They have spliced animals and humans together. They're now treating humans like animals. In this particular study I just mentioned, it says they are using worms as carriers for the messenger RNA. They are transfected first with the green fluorescent protein, insect DNA, and then they are the vectors. And in particular, they are targeting the REX1 on the X chromosome for deletion. What this does, to answer your question, is it causes their offspring, if they can have children, and there's a very small percentage you can, and that's specified in the study, their offspring are mutants. That's the exact word they use.
Who's they?
And
Who uses that word? Who says that they're mutants?
NIH.
Tony Fauci's NIH.
It's right there on the website. Yeah, absolutely. And it says also that they can create a hermaphrodite sex by deleting this REX1 on the X chromosome. So they can play with the sex of people and of the next generation. They can make people completely without sex. There's one more study I wanted to mention on the NIH website. It's entitled Core Cross-Linked Fluorescent Worm-like Micelles for Glucose-Mediated Drug Delivery. These micelles are worm-like. These are the hydrogels made from graphene oxide for drug delivery that have been found on the PCR swabs, on masks, etc.
They are deleting God's creation is what they are trying to do. There's the studies right there at the NIH website. They are telling you, Tony Fauci's NIH is telling you that they are cloning you and turning your children into adronomous mutants. Insane.